Right. So thank you very much. So yeah, this is the first lecture in the new series of this set of 4 lectures on ocean ecosystems and climate. So I'm Thomas Jackson from the Plymouth Marine Laboratory, and uh, although I'm giving the talk, there's a huge amount of work in here. I think if I put the full list of people that have been involved in all this work, it would fill probably more than a full slide. But all the references that I give for the work and things I discuss, I'll provide at the end in a big list. So you can go and do further investigations and read a bit deeper into all of the material. So quick overview of this module, this set of 4 lectures first, and then I'll press on with the main body of this lecture. So the next 4 lectures we have ocean colour and climate, primary production, phytoplankton community structure, and indicators of ecosystem status. So this lecture is on ocean colour climate, and that I'm going to mostly focus on what it means to have a long data record, how you generate those climate quality data sets, and then give a few examples of using those for what we might consider climate change or climate investigations. The second lecture following this one will be from Bob Bruin, and that's on the carbon cycle. And so he'll talk about the carbon cycle from the perspective of ocean colour, and also look at biological carbon fixation in the ocean. So just a little teaser for that. It's quite a complex system. There's a lot of different pools of carbon in the ocean, and they have it's got quite a rapid turnover from the biological side. Which he'll go into a lot more detail of. But anything where I mention primary production or carbon fixation, you can expect to get more information in next week's lecture. The third lecture will be from Dr. Heather Bowman, and that will be focusing more on the diversity. So once we've looked at climate first and then the carbon cycle, then we'll drill a bit further down and have a look at the diversity in the size and the function and the pigmentation and characteristics of plankton that make up this community structure and how that varies across the globe and in different environments. So one part of that is that the algae have different colors, as you can see in the bottom figure here with the different vials. But also looking at things like the enormous range of sizes that you get in phytoplankton. So from things like the cyanobacteria, which is Prochlorococcus here on the left of the top figure, through to things like Trichodesmium, which are colonial, there's 4 orders of magnitude or 5 orders of magnitude in size. And that's the same as going from a single fish to the city of Manhattan. So it's really, you can't consider these organisms often as a single type. We have to segregate them in different ways. So then the last one in the series will be from Dionysus, and that's going to be looking more into, once we have these records and we understand what's going on in them in terms of carbon or in terms of phytoplankton groups or functional types or biomass, how do we extract a little bit more information from those records? So that will be things like key metrics or phenological indicators and getting a bit more information out of the time series records. So here's an example from some work that Dio did with Marie-Fanny Racot, and this was looking at the Red Sea. And you can see in the left side of this, we've got just regional extractions of time series. The red is temperature and the blue is chlorophyll. And you can see that they have some seasonal cycle across the year. And then when we talk about phenology, what we're doing is we're trying to extract metrics which represent those sorts of features in time. So that might be the duration of an event, the peak amplitude of an event, or the timing of an initiation of an event. So Dio will dig a lot more into that and also relate that on to things like fisheries and higher trophy levels. So back to the oceans and climate, which is the main topic of this lecture. So the Earth is a blue planet, fundamentally. It's 2/3 covered by ocean, and around 97% of all Earth's water is in the oceans. And the oceans play a key role in regulating the Earth's climate. So not just in terms of them being a huge source or vector for temperature, well, heat moving from the tropics to the poles, but also with things like dissolved gases. They really are kind of a buffer and a key player in the whole system. So over the last 50 years, although there's been a lot of warming and there's been quite a lot of interest in, say, air temperatures over Europe or Africa or other areas of the globe, 90% of the excess heat from the warming of our planet has actually gone into the oceans, not the atmosphere. And during that time, they've also absorbed around 30% of the anthropogenic CO2 that's been emitted. So without the oceans having this buffering effect, temperatures would have risen much more in the atmosphere and the CO2 levels would be even higher than they are now. Additionally, so beyond just the physics and chemistry of the gas exchange and the heat transport, phytoplankton in the ocean are responsible for roughly half of the primary production on the globe. So they are equivalent to all of the terrestrial plants, basically. I believe Shuba mentioned this before. And so any changes in the physical and chemical ocean variables can impact on the marine habitats. So we know that things like increased temperatures or increased acidification of the oceans have direct impacts for things like coral reefs, but they can also have impacts on other marine ecosystems and marine resources. And we know that there can be large variations of phytoplankton production in the geological record. And in fact, when we think about carbon on very long timescales, on geological timescales, pretty much all of the oil that we're currently extracting as a fossil fuel originally is sourced from plankton that were buried in the oceans. And this is a beautiful picture on the right here from Meteosat showing Africa and Europe and a large section of the Atlantic. But I'd also like to just show another image from the Pacific, because this is from the European or the Eurocentric view is one that many kind of have stuck in their mind as a vision of Earth. But really, if we look at it from the Pacific, you can have a hard time here trying to find where the land actually is. So you can just about see California over on the top right here. But really, this is much more of a representative picture of the blue Earth. So the Pacific-centric view, really showing how much of the surface is covered by ocean. So before I really dig any further into that and we start to look at how we might create climate data records, I think whenever we're asking questions, and we've seen this through all of the lectures in this series so far, where we have questions coming into the chat as we go through and also in the Q&A session at the end. We have to think about how we form questions and then how we answer them. So the scientific method in general starts with questions. We have a question that we don't know the answer to. We have to go and gather information, form some sort of testable hypothesis, because if it's not testable, we can't get a truth or increment towards the truth with our investigations. Usually that involves designing some sort of experiment, or if not an experiment, some exercise as a testable objective. And then you go out and you test your hypothesis. And once you've got your data and you've analyzed it, ideally in an unbiased manner, you interpret that data and form conclusions that you can share with the rest of the community. And often that's the point at which you just have more questions. And that's part of the joys of science is with every answer, sometimes you have another thought and another question. And you can go around that loop a number of times. Ideally, you can publish your results and then your results can be retested by the rest of the scientific community, adding credence and validity to those results. So really, by joining and coming to these lectures, you're already in part of this flow. You have questions, you are consulting experts, and through the lectures that we give, seeing summaries of the literature and the current state of the art. And hopefully that's answering some of your questions. So then you're going to the yes, and you can go and use that newly acquired knowledge. And ideally, when we get to the end of this whole series, you'll feel empowered to go and form new questions and really push things forward in your own areas of interest. And ideally, that means you'll end up going down the no path here. You'll have a question that we haven't been able to answer, but we've tried to provide the tools for you to answer that yourself. So in that case, you would then follow on down the path of working out how you might test your question or test a hypothesis about a question and get to some results. And we'll go over that process later with a few worked examples in the climate realm. And really, I think I would like to view this lecture series as just helping you get expanding your sphere of knowledge relative to the total sphere of knowledge. So this is just an aside, but for me, it's how I visualize the learning process. If we consider the whole sphere of human knowledge, at any point within that, you can consider starting at the center, knowing nothing. And as you learn more, you progress towards the edge of understanding, so that edge of that sphere. And the direction that you're traveling in from that center is just a different topic. So whatever you're learning about, you're pushing out towards more and more understanding and the limits of that knowledge. And once you reach the edge, once you get to the edge of that level of human understanding, you work and you push and you investigate, and hopefully you push it out a bit further. And if everybody does this across all of the fields of science and investigation, then the whole sphere grows, increment by increment, piece by piece. But this is not always a linear path. In fact, it's very rarely a linear path. Normally, you're learning about one topic, say ocean color, and you find out you have to go and learn about something else, perhaps Fourier transforms or sediment dynamics or atmospheric processes, or I don't know, light propagation. There's all sorts of other little things you want to go and learn about. And eventually that brings you to the point that you need to be at the edge of human knowledge. And through my experience, normally that's the point at which you really have to take another deviation and you'll pull in information from a different area of science or from a conversation with someone who works in a related field. And that will help you make that bounce, that step out and push knowledge a bit further. So anyway, with all that in mind, about how we ask questions and how we make progress, back to ocean color. So ocean color as an indicator. Really, this has been mentioned a number of times in the introductory lectures from Lauren and from Anna. But so phytoplankton are sensitive to their local environment. And the figure on the right here shows a lot of connections basically between phytoplankton and bacteria and light and nutrients and all sorts of ocean processes. But the most important fundamental drivers for phytoplankton growth are going to be light and nutrients because they're photosynthetic organisms. So they need light in order to photosynthesize and they need nutrients in order to synthesize new organic matter. Once they have those, then factors like temperature or pH or salinity, the presence of grazers or sediments which might block the light and things like that, will also have secondary impacts on their ability to grow and flourish. But what that means is when they are growing, and we can see them because they have this optical signal, they can be reflecting invisible changes that we couldn't see. So we don't yet have the ability to map some nutrients from space like nitrate or silicate. But if we see particular phytoplankton growing in abundance, then that's probably due to a response that they have to the change in the nutrient levels. So they can be used as a proxy. And we've talked about proxies in previous lectures. So in that sense, the phytoplankton themselves can be an indicator of other change. And there'll be more in the later lectures on phytoplankton primary production and the carbon cycle and how that all links into the hydrotrophic levels. So I'm not going to go too much into the detail on that now. But it's also worth noting that because they have this foundational role in the food web, that any changes we see in the phytoplankton dynamics, so whether it's in their abundance or even which type of phytoplankton are growing, it can have a significant impact on the rest of the marine ecosystem. So sometimes it's useful to think about phytoplankton as these sentinels of change. So they are responding to things that we might otherwise not be able to see, but also they're important. And when they change, we might see knock-on effects later at other levels within the marine ecosystem. And there's just a nice, so this plot on the right is from a study in 2003 by Trevor Platt and colleagues, looking at the spring bloom in the North Atlantic and seeing when that bloom is shifted in time, so early or late, what's the impact on survival index of fish larvae. And so what this means is basically if the fish larvae aren't lined up in time, they're not there at the same time as the phytoplankton bloom, they have a hard time getting enough food to survive. So there we see a knock-on impact from changes in the phytoplankton to the higher levels. And I don't think the plot here on the chart on the right here has been shown in previous lectures, and this is a really nice summary from a paper from Trevor and Schuber that was looking at a range of indicators that you can derive from ocean color. So when we talk about ocean color in the climate record and things like that, or as an ECV, which I'll come on to later, primarily that's reflectance and chlorophyll are the variables, but from that we can derive a whole number of indicators. Some of those are phenological, such as the ones at the top about timing of a spring bloom or the amplitude of a bloom. Some of them are more variable based, such as the particular carbon or the phytoplankton carbon. And some of them are rates, like growth rates or loss rates. So there's a whole number of indicators that we might be interested in when we're trying to look at ecosystem status and function on longer timescales. So let's come to a broad question in our kind of schema of forming questions and testing hypotheses. We can imagine that we might have a question about climate change and the marine ecosystem. So is the climate change that we see going on and how that's changing in the oceans having an impact on the marine ecosystem as a whole? But even here, we have to take a hard look at this question and think about what we're actually asking. So what changes do we think of when we say climate change, and what's classed as a change in climate? And an important factor on that is what data should I use when I'm looking at climate change? When we talk about impact, what do we mean? Having an impact on the marine ecosystem. And are we talking about the whole marine ecosystem, or are we talking about a small part of it? And none of these, if we say we only want to look at part of the marine ecosystem, that doesn't mean that that's an invalid question, but we just have to phrase that question carefully and clearly when we come to forming a testable hypothesis. And an important part of this, a huge part of this, is that you have to make sure you use climate quality data whenever you're trying to address questions about climate change and climate change impacts. And I'll get onto what climate quality means, but it's quite stringent. So first, let's think of those 3 points that we were thinking of: climate change, impact, and ecosystem monitoring. So climate change is over long timescales, usually from decades to millennia. This is not about whether the level of chlorophyll in the ocean is higher today than it was yesterday. This is about what's happening to the region that you are looking at over much longer scales. So there's a whole number of variables we might consider when we think about climate change. And there is a group called the Global Climate Observing System, summarized as GCOS, which have defined an essential climate variable as a physical, chemical, or biological variable or group of linked variables that contributes to the characterization of the Earth's climate. And they have got a whole list, a whole number of different ECVs, so these essential climate variables, that they have separated by basically atmosphere components, land components, and water or ocean components. Now, I'm not going to go into a huge amount of detail on all of these, but just know that whenever I talk about requirements or quality, or I show you the level of detail that's required for an ocean color ECV, the same is true of all of these. And this is why when I say when you're thinking about trying to look at climate studies, you have to make sure you're using climate quality data. And that could be temperature of the ocean, it could be temperature of the land, it could be properties of sea ice, or all sorts of things, but you want to make sure you're using climate quality data sets. So impact. When we talk about impact, normally we're talking about a change that is detected with a given level of confidence. So if we see a change and we can say we are 95% confident that this change is not just an aberration of noise or anomalies, this is a significant trend or shift. And also, when we say impact, it might be that you have one primary impact, but you could also have knock-on effects and feedback loops. So we have to think about those when we talk about the climate system. And the marine ecosystem, so rather than considering the entire ecosystem at the minute, just for this talk, I'm going to focus with ocean color down onto the primary producers. So the phytoplankton, which are a foundational layer. And as I said, because they produce about half of the world's oxygen and support all of the higher marine ecosystems, it's actually a very important layer to look at. So if we see changes in that component of the ecosystem, we would expect to see changes elsewhere. So just to show on the diagram, if we zoom in onto the ocean section, you can see ocean color is down here, highlighted among the group of ocean biogeochemistry. It's also worth noting that ocean color is the only ECV here, which actually allows you to look within the ocean biogeochemistry section that allows you to look at what's going on biologically. The rest of them are often related to inorganic or chemical processes. So ocean color as an ECV or CDR. So the other term you might hear when we talk about climate records is climate data record. So ECV is the terminology used by GCOS and ESA. And so that's the essential climate variable. And GCOS defined a set of requirements after discussions with the community around what would actually be needed from ocean color for climate studies. And they have these requirements set out for all of those ECVs that I just showed in that diagram. So if you're interested in a different variable, you can go and look up what the community requirements for that were. But for ocean color, we have a spatial resolution requirement of at least 4 kilometers or better. Temporal resolution should be daily. We can then create composites, say a weekly or monthly, but fundamentally we need daily resolution. There is a requirement on measurement uncertainty. So that is 5% for the reflectance product and 30% for the chlorophyll products. And a requirement on stability. So long-term, basically what that means is that the processing system that you're using to generate the variables is not introducing any trends. So things that you see as a trend, are coming from real change and not from an instability in your processing system. I'll get onto that in a minute. So a climate data record, it sounds very similar. So a climate data record is a time series of measurements of sufficient length, consistency, and continuity to determine climate variability and change. So sometimes there is a little distinction between CDR and ECV, where CDR is the more, let's say, fundamental or primary record. So that might be reflectance, whereas sometimes you might consider chlorophyll as being derived because we use the reflectance to calculate the chlorophyll. So the CDR sometimes is thought of as reflectance. So anyway, with that in mind, so we have a definition of ECVs and CDRs, and we know that they have to be very high quality. So the European Space Agency, a number of years ago, set up the Climate Change Initiative. And that was a project, or is a project, it's still ongoing, that was designed to create these ECV records using satellite data. And if you go to the link here, you'll see there's a whole list of ECV data sets. And one of those is ocean color. So we'll go into that in a bit more detail now. But I just want to stress again, these sorts of, when you hear things like measurement uncertainty, stability, consistency, continuity, that's the fundamental properties we have to have in climate quality data. So this is the full set of ECVs from ESA. You can see they span different amounts of time. That usually depends on which sensors were available to create the record. You can see again they're split by atmosphere, ocean, and land groups. Within the ocean groups, the top one you can see here, this is ocean color. But if you're interested in any of these, this is a really good place to go and look for data sets. And actually, it's really nice. Often you'll find the really interesting science is when you start merging multiple data sets into analysis. So if you look at, say, ocean color in relation to sea ice or in relation to temperature or other things. So we'll get onto that in a minute. Good place to go looking for data. So what goes into generating an ocean color CDR or an ECV data set? So this figure, you've seen something like it before when we had the introduction to ocean color, and I don't want you to have to understand right now every term in here. It's intentionally, it's a complex equation, and that's because the atmospheric correction stage is tough. It's hard to do. We're trying to extract a signal which has a lot of other things in there that we don't want to see. So if I just skip on here, so on the left we've got LT, which is our top of atmosphere detected radiance at the satellite, and right over on the right hand side you can see the LW, and that's the bit we actually want from the water. Everything else in the middle is things that we have to remove or correct for during the atmospheric correction process. So we've got path radiances, which is about 90% of the signal needs to be removed. You can think of other things that might contaminate the signal, such as whitecaps. So when the surface of the ocean is whipped up by wind, we've got specular reflection, so glint from the sun, and we've also got to take account of the atmospheric transmittance. So there's, to dig into this, obviously I'm happy to answer questions, but if you want to do more independent reading, I would recommend going and having a look at the IOCCG reports for all the fundamentals of ocean color, but also the Ocean Optics book has a lot of useful information on solving radiative transfer equations and things like that. So really good resources there. This is just one I wanted to highlight because it's a really nice example of what we mean by corrections. So when we talk about the LG term, so here in the middle, you can see this specular reflection, LG. That can be both direct sun glint, which I'm sure anyone who stood next to a body of water on a clear day has seen sun glitter or sun glint, this specular reflection. And this is a problem for satellites because it's extremely bright. It used to be that we would just mask out any area that from the viewing geometry we thought would be contaminated by glint. But actually more recently, the atmospheric correction schemes such as polymer have become quite advanced and allowed us to push into that glint region, and they can actually calculate and resolve that glint component and remove it properly. Another component to the surface reflectance that could be interpreted incorrectly is if you look at the image on the right, if you ignore the sky, you might think that there is a change in the ocean properties between the left and right side of the image. The left side of the image, the ocean looks grey, and the right side, the image looks blue. But actually, this is not a change in the properties of the water. This is the reflection of the skylight. So again, this is a component that we would be in error if we tried to attribute that signal to in-water properties. So again, this is just me highlighting some of the difficulties that we have to make sure we've accounted for properly when we're looking at high quality data sets. So back to that equation, if we assume that our processing works and we've managed to correct the path radiances, the glitter or glint radiance, the whitecaps, and we understand what's going on with the atmospheric transmissivity, then we can calculate the water leaving reflectance. And that's what then constitutes the ECV, or the ocean color record. So we have this record of data that we've processed, but how long a record do we need? How big does the time series have to be for us to detect climate change? There was some nice work done by Stephanie Henson and others and they actually used a model or a series of models to try and determine the timescale you would need to have constant observations in order to detect a climate change signal. Now, fundamentally, you can think that it's obvious that if you have oscillations on the period of a year, you definitely need more than a year's worth of data to be able to detect any significant change. If you have oscillations on a period of 10 years or 8 years, like the El Nino cycle, you need a time series longer than that to be able to detect longer term shifts beyond that natural variability. So fundamentally, for any time series analysis, the duration has to be longer than the natural period of system variability. If we dig into a bit more detail, the properties that really determine that length of time series required. So there's the standard deviation of the noise. So the noisier the signal, the longer the time series we need. There's the magnitude of the trend. So if you have a very strong trend, you don't need as long a time series to detect that trend. And there's also autocorrelation of the noise. So autocorrelation is where your time series has a correlation with a later time step. So you might imagine if you were taking measurements of a child's height through time or something like that, obviously that's always going to be increasing because we know that from time step 1 to time step 2, it can only go one way. So there's an autocorrelation there. So by accounting for those autocorrelations, or having to account for those autocorrelations, we'll also change the length of time series required. The last point to consider, it's actually very important, is gaps in the record. So if you have a time series of, say, 10 years, but you measured 5 years, and then you have a gap of a year, and then you get 4 years of data, that is not as useful as having 9 years of continuous data with a gap at the end. The gap in the middle of the record detaches or breaks the continuity of that record and means you need a longer time series to be able to state that the trend is real and that you know it beyond a certain level of confidence. So looking at the maps on the right, the two columns, so one is chlorophyll and one's primary production. We don't really need to make a distinction between those at the minute. And then the 3 rows are just different model types. So there was a number of different models used in the exercise. And the different models showed slightly different patterns, but we can see overall there is a variance between around 20 years of data and 60 years of data needed to be able to detect a trend in most of the oceans. The Arctic is one region that stands out as needing a shorter record. And then as we move into areas like the gyres, which are very stable, and clear blue and have a low cycle, seasonal cycle, low noise, and those have very long periods required. So we need at least 20 years in the shortest areas to detect any sort of climate trend. And there isn't a sensor available that provides that length of record on its own. We haven't had a satellite providing good data for 20 years. So what we need to do is merge sensors. So here's a list of ocean color sensors. Some of them are no longer running, such as CWIFS and MERIS. And also, I mean, I would consider MODIS now not really operational. It's a dying sensor. So current sensors on here, we've got Sentinels 3A and 3B. And then into the future, you know, we would expect to see PACE and Sentinels 3C and 3D. But hopefully you also notice with this list that if we look at the properties of these sensors, so whether it's the spectral bands, whether it's the resolution, spatial resolution, also those spectral bands have different sensitivities or different spectral resolutions. All of that changes from sensor to sensor. And on the one hand, that's good because the capabilities of the instruments that are being launched is increasing and improving, but it makes it difficult when we're trying to merge these sensors into a single record because we don't have the same data coming for the whole 25 years or so that we have so far. And we have to try and account for that and make sure we don't introduce artifacts where we've got different sensors contributing to the record. So one of the added benefits of merging sensors to get the time series that we need of 20 or 25 years is that we also increase the coverage. So the top left, bottom left and bottom right panels here show individual daily composites from 3 different sensors. We've got MERIS and CWIS and MODIS. And then in the top right, you can see the merged data set. So not only does our time series grow in length by adding these sensors together into a merged record, we get much better coverage, and that means there's also less gaps in the record. So it's a win-win. So we get multiple benefits from merging the data sets. But we have to be extremely careful. So in the top figure here, we've got 6 different sensors listed, and you can see they all have slightly different wavelength sets. The closest pairing is probably MERIS and Sentinel-3, but even there, Sentinel-3 has bands that MERIS didn't. And to have a consistent record across the entire time series, we basically need the same wavelengths to be measured from the start to the end. So what we do within OCCCI is we have this processing stage that we call band shifting. So we pick a reference sensor, and for the latest versions that has been designated as MERIS, and we correct the data that we have at the native wavelengths of each sensor to the wavelengths of the reference sensor. I can go into more details perhaps in the Q&A if people are interested, but basically this involves running a forward model to generate a hyperspectral signal, and then we extract from the wavelengths that we actually want for the reference sensor. But we also have limits on how far we're allowed to shift. So we don't want to be trying to shift, say, from 670 nanometers up to 720. Like it's too big a spectral jump. So we're only allowed to shift a certain distance spectrum. So that's the band shifting. But even when we band shift, that isn't the whole story, because even so if we consider 2 different sensors in orbit, even when they have the same wavelengths, those 2 sensors are not identical and they aren't seeing things in an identical manner. So if you look at the plot in the bottom left, that's an extracted data set for an area in the South Pacific Gyre. And the South Pacific Gyre is a very stable region of the ocean. And so you can see there's a series of sensors on there. We've got CWIS, MERIS, MODIS, VIIRS, and the 2 ULCHIs. Now, you would expect the 2 ULCHIs to agree relatively well, because by design, those are supposed to be identical sensors. So you can see the pink and the red overlap pretty closely. But all of the other sensors have significant offsets. And if you imagine taking something like the MODIS record in purple at the top and the ULCHI record in pink at the bottom, and trying to merge those or average the 2 together, when you introduce ULCHI, you would see a big step in the record. And we don't want that. That's an artifact of the merging process rather than a real representation of a change in the system. So we have a what we call bias correction step, which accounts for the differences between the different sensors. And that's done for every single pixel on the globe every day of the year that we have measurements for each sensor. But when we constructed that methodology, we have to do it in a way that it cannot introduce trends itself. So we don't want to have a method that might artificially create a downward trend or an upward trend in the record as it's attempting to correct between the satellites. So what we do is we use the overlap periods between sensors to basically create a daily climatology of the offsets between them, and then we apply that back as a correction. And that gives us the result on the bottom right. And you can see there pretty much all of the sensors are agreeing in terms of the magnitude of the reflectance, the seasonal cycle of the reflectance. The one thing that stands out is we've got a little bit more noise during, or higher range during the CWIS period. And so obviously that was an older sensor and maybe didn't have the quite the sensitivity that the other sensors have, or the number of bits available in its reflectance bands. But yeah, overall we're happy with that band shifting and bias correction process that is giving us a good ocean color record. So as well as systematic anomalies like that, we also have to take account of transient anomalies. And there's a whole number of sources of these that might come up. And this is part of the QC and quality control and checks that we've iterated over the years within Ocean Color CCI. So if I just draw your eye to the circles in the lower panel here, these images are taken, well, the daily composites taken one day apart. So we have the 7th and the 8th of April in 2005. And you can see this is reflectance. You can see the reflectance changes significantly in these two areas from day to day. And you might imagine from what you know or have learned from this course about the cycles and changes in ocean color and ocean properties, this is a very big change to happen in one day. And I haven't put it on here, but if I showed you the next day, they would actually revert back to values like this. So it's flipped just for a day. So if you think for a second maybe about what might have caused that, you know, there's all sorts of things it might be. But when we dig into it, what we actually find out is that the image in the top, when we go and look at the true color, looks like this. So it's got blue oceans and some white cloud. Yeah, that makes sense. This is what we expect. Down here, the ocean looks almost black and the clouds have gone kind of brown. And this is puzzling, clearly not what we would expect to see in an ocean color image. And it turns out this is a solar eclipse. So it just so happened as the satellite sensor was taking a picture of that part of the ocean, the shadow of the moon was also passing across. And this down here, so the moon transited. So as the satellite went over and came around for its next orbit, it picked up the shadow again in a different location. So this was like a double hit from a single solar eclipse. And these are the sorts of things that you also have to be very careful that you have cleaned the record and made sure it's top quality before you're doing climate change studies. Now, if we go back to the lecture that Lauren gave first, she really stressed the point that you can't have everything all the time everywhere. You have to make a choice between whether you want high resolution, you know, revisit time, spectral resolution, sensitivity, signal to noise ratio. All these sorts of things are properties that you have to balance. You can't have everything. So when it comes to the climate record, consistency and traceability and sensitivity, these sorts of things are very important. And it means that sometimes we have to push coverage down the list a little. So here is obviously we would want to mask the solar eclipse. That's definitely going to be bad data and corrupt the record. There's also steps where we have to make a choice, and it's sometimes it would be nice to have the data, but we have to apply the masks because consistency and quality has to be our primary goal. This is an example of just that. So in the left image, you can see there's much more data than in the right image. And the reason for that is just one of the many flags or masks that are applied during the processing has been turned off. So the difference is that there's no stray light mask, and the stray light mask is designed such that around bright targets, you can have a problem where that bright object, the reflectance from it is scattered within the atmosphere and contaminates adjacent pixels. So basically around things like clouds and bright objects, there is this mask. In this image, you can see that by removing that mask, you actually get a lot more data, and it looks reasonable. It seems to follow the same patterns and shapes that they look oceanographic, they look realistic. So you might think, let's turn it off. And we've seen examples of people trying to generate climate quality data with this turned off, and it's a problem. Because you go and process an image somewhere else, and this is the difference. And you've got a whole bunch of horrible data, which really shouldn't be in the climate record, all around the clouds. And in some ways, you might think of this as being noise, but actually, because this is around bright objects, this actually has a slightly systematic component. It will tend to influence things in the same way. But anyway, that's just a thing. So we always have to stress quality and reliability and traceability. So if we provide chlorophyll, you would expect that you should also see the reflectances to go alongside it, because if the reflectances aren't good enough and you don't trust them, you probably shouldn't be deriving chlorophyll from it. So once we have a high quality reflectance data set, we have to generate downstream products like chlorophyll. And there's been a whole bunch of questions over the past weeks around which algorithm's best, which algorithm should I use? And at the global scale, the answer is there isn't just one. There is no single algorithm that performs best across all waters. So when we're merging all this data together, the approach that we've taken is to use an algorithm blending approach. So the way this works is that we look at the spectral shape of each pixel, and we assign it a membership to a number of different spectral types. And those are shown in the plot on the bottom left. And we know, or we can use in situ measurements to say which of the algorithms perform best in each of those types. So then when we have the memberships to all the types, we can weight the algorithms accordingly. So then if you look at the image on the right, the top image is the output blended product. And the idea is that this blending wouldn't introduce artificial boundaries. You don't get a hard line where we flipped from one algorithm to another. It's a smooth, continuous process of weighting the algorithms that are doing best in those optical conditions. And then, so if we zoom in on the box area, you see the four lower figures, and that's the blended product, and then basically the contributions from 3 different algorithms which are going into that blending. But the final product is smooth, and you can see that the OCI algorithm is mostly used offshore, whereas the OC3 and OC5 algorithms are mostly used well, the OC5 is used closer to the shore in this example, and that's the final output product. So it's not just one algorithm, it's a blended approach. And so when I was saying about finding these bad pixels, it takes a lot of rigor and a lot of effort and a lot of checking to go through the climate records. And in some ways, we rely on we do a lot of that internally within the project, but it's also really useful to hear back from the community when they've found things that are odd. And that's gone on over the last decade within OCCCI, because basically 1 in a million happens all the time when you're processing billions of pixels. So the y-axis on this chart, the values 0 to 5, that is 500 million pixels, or 100 million observations processed. And this is daily through our record. So you can see when VIIRS kicks in, that's 400 to 500 million observations per day going into the total record, added to all the other satellites. So it really is billions and billions of observations. So if there's a condition that could be tricky, we will have to deal with it at some point. Now, I've talked about systematic errors and systematic issues like intersensor bias correction, and we've pushed more into the kind of noise or the random errors that can be a problem, and that's often this 1 in a million type. And which of those errors is more important actually depends on the scales that you're looking at. So if you're looking at short time scales and small distances, the random errors are much more important. They will potentially corrupt your record. As we move to coarser resolutions, degree, 2 degree, 4 degree composites, and long time scales looking over decades, they tend to average out, and the systematic errors become much more important. So it's just something to be aware of, that depending on the spatial and temporal timescale that you are interested in, you have to pay attention to which errors are more important. As creators of a climate record, we have to care about all of this, because we don't know if our users are going to be doing stuff with 1 degree composites, or if they want to be looking at 1 kilometer or 4 kilometer data for their local region. So after all that, the OCCCI project has this processing system which ingests data from multiple ocean color sensors. It goes through atmospheric correction. We have all the band shifting and bias correction. We have algorithm blending, and we output products, and those also have associated uncertainties, which allows them to be ingested into things like climate model analyses. And this is part of a cycle. So we've gone round about 6 times now. We've had 6 dataset releases, and plans are afoot for future datasets. And we take it as a sign that the data has been good quality and taken up well, because we've always had good feedback. There's been over 180 publications now that have used OCCCI data. And it's also gone through into really high level stuff like the IPCC working group reports, and the Copernicus Climate Change Service, and the reports that have come from that as well, European State of the Climate Report. But the cycle continues, it goes on. And I will just take 1 minute to say that part of that process, that validation and quality control and understanding how good the products are, it relies on good in situ data. We've heard this in a number of lectures, so I'm just reinforcing the point, but we cannot overstress the importance of in situ data for grounding the satellite data records. So within OCCCI, there has been a compilation made of ocean color in situ data. So that's got reflectances and TSM and chlorophyll and absorption by phytoplankton and backscattering. And that's publicly available. So I've linked to the paper there, and the data set's on, I think, Pangaea or ESSD. And you can take that and have a look at it yourselves, download it, have a play with it. And when we do that analysis for the OCCCI products, this is for chlorophyll, we get a pretty good relationship. You can see here that the bias, overall bias, which is the delta in here, is very low, and that's good. That reinforces pretty much what we were trying to minimize with any biases in the data set, especially over long time series, over the long time record. So yeah, we have a pretty good performing global chlorophyll product over the 25 years or so of the record, and it looks something like this at the long time scale. So you can see the blooms in the northern and southern hemisphere. You can see local dynamics of upwelling off the coast of Africa. You can see the Amazon plume and all these sorts of things. And over the long time scale, this is the global median chlorophyll at the bottom. You can see that evolving over time. So we have this data set, and then we have to use it. Also, for a bit more information, please go and see all these various places for either access to the data, or there's a few useful videos, kind of introductions to the CCO project, all the different data that's available, and why it's important. So I'll put all these in the thing at the end. So coming back to this original question, we have a climate data set now that we're happy to use. So can we actually see if there's any changes going on? And I'll speed up a bit looking at the clock. So we know the Arctic is likely to be an area where there's we need to detect. We would be likely to detect some change. We saw that in the work from Stephanie Henson. Basically, that's where if you're going to try and find something in a 20-year record, that's a good place to look. We know that phytoplankton respond to light stratification, forcing changes in nutrients. We know where we can get some good quality data now. So maybe we should look in the Arctic at ocean color and see if there's a shift in the record, and if so, what might be causing it. And rather than just saying all latitudes, high latitudes, the whole Arctic, we're going to focus on a small region just for this demonstration purpose. And there's a nice example of this. It was a published paper from Andrew Orkney, and the premise of that paper was that there's ongoing rapid change in the Arctic. We know sea ice extent is falling. We know sea surface temperatures are increasing, and we know the amount of energy in the atmosphere is increasing as well. Potentially it's becoming more stormy or turbulent. So we might expect that with those changes, we would expect to see changes in either the composition or the phenology of the phytoplankton. And those are pretty significant changes, and there's likely to have an impact from what we know around the drivers of phytoplankton growth. And there was a recent review that noted that autumn blooms have been occurring in the low Arctic as a new phenological feature. So Normally in the Arctic, it was classed as primarily just one bloom, rather than this spring and autumn bloom. But now we're starting to see spring and autumn blooms together. And again, this is coming back to this list of indicators. So we're looking at what might be changing and which indicators we might use to look at that. So after a literature review of the area and what might be changing and driving phytoplankton growth, there were 4 candidates for what might be facilitating these autumn blooms or increase in the autumn bloom frequency. It could be more mixing from the wind. It could be water column destabilization from Atlantic inflow. It could be nutrients coming in from the Atlantic, i.e. more nutrients coming in and promoting more blooms. Or it could be that there's enhanced nutrient replenishment from mixing over bathymetry, kind of seabed features. So the hypothesis to test basically is that if the inflowing Atlantic waters are supplying nutrients to support these autumn blooms, then a clear relationship between the Atlantic inflow and remotely sensed chlorophyll would be expected. Okay, I have a nice hypothesis to test. So in that study, they went and they looked at a number of variables. So in the top left here, we've got the trend in chlorophyll. In the top right, we've got the trend in sea surface temperature. In the bottom left, we've got the trend in windiness. And in the bottom right, we've got a trend in the currents. And this has been regridded, I think, to 1 degree, and they've taken only September data across a 17-year record, which I think is the record that was available for all the variables. You can maybe see there's little circles and triangles. The difference between those is to do with autocorrelation. Again, I'm not going to go into detail on that now, but basically all the points that have either a circle or a triangle were classed as significant trends, so statistically significant. And what you can see is that basically the chlorophyll and the temperature in September are increasing pretty much everywhere. There's nowhere where they're really decreasing, whereas the patterns in the windiness and the current speed are a bit different. You could say that the chlorophyll is maybe increasing more in the southwest and not so much at the highest latitudes, whereas temperature is increasing pretty much everywhere. And if we look at a time series of these across the region, so you can see the chlorophyll in green, we have the SST in orange, the currents in black, and the windiness in blue. And the strongest agreement of any other variable with chlorophyll is for the currents. So this is the black line and the green line. So although energetic winds do promote mixing, the interannual variance doesn't agree with the change that we see in the biomass, but the current speed does. So although there is a hint of a relationship between the bloom biomass and the surface temperature, that's actually more likely due to them both being forced by the Atlantic inflow rather than there being a direct forcing from the temperature to biomass. But an interesting side note on this or point from the study was that although here it looks like an increased Atlantic inflow or Atlantification, as it's termed, is pushing the biomass for the autumn bloom up, it might be that or it seems that there have been studies to show the Atlantic nutrient inventory has been decreasing. So it might be on longer timescales, we might see this change reverse because additional controls on the nutrient provision might actually dominate. So we'll have to wait and see. But in summary, basically this study has gone through that whole process that we discussed right at the start about forming a question, getting some relevant data, creating a testable hypothesis, analyzing the data in a clear manner, and presenting the summary results and the conclusions. And basically here it's that the strongest support was for this increased nutrient inventory from the Atlantic, whereas for the others, we didn't see as such a strong support for that hypothesis. So moving on to a second, just a quick example here that, and the reason I put this one in is I think it ties in nicely back to some of the work from module 2, where we saw Grinson talking about the mini Secchi and water quality indicators. So in those lectures, we were told things like the Secchi depth, the hue angle, the Forel-Uhl index are really useful indicators of water quality. So can we look at these as a climate record? Can we take our climate record of ocean color and try and look at water quality across those scales? So there was a study from James Patak and others, which took the OCCCI data as input, developed some algorithms to calculate Secchi depth and the feral ool index, and then performed some trend analysis on that. So the maps that they produced of Secchi depth, hue angle, and feral ool can be seen here. I wouldn't worry too much about the standard deviation or coefficient of variation right now. If you want to dig into it more, go to the paper. But you can see in the mean panels in the top that they make sense. The deep blue oceans, the gyres, have a very low FU index. The Secchi depth is very high or deep compared to the coastal waters and the Arctic, for example. So then what they did was they took those products and analyzed them across the time series, so the 20 plus years of CCI, to look for trends. And so here you can see a panel for the North Atlantic. The columns left to right, P1 to 3, are 3 different areas shown here. So we've got the Northwest Atlantic, we've got the Central Gyre, and we've got the Eastern Atlantic. And you can see if we look at the Secchi depth on the top and the hue angle on the bottom, they have potentially different trends in different areas. And that's just for 3 individual pixels. But then if you look at the trend in the red line here, which is the deseasonalized values. So there's a seasonal cycle, and then they've deseasonalized it to get the red line. The trend in that for each pixel is shown in these 2 panels. And you can see it looks like there's an increasing Secchi depth in the central gyres, with the waters becoming clearer, reflected also in the change in the hue angle. So going back to our big question that we posed a long while ago, sorry it's taken so long to get all the way back to this, but if we look at climate change in the oceans and if it's having an impact on the marine ecosystem, well, we know that the oceans are facing pressures from multiple angles. There's ocean warming, there's acidification, there's pollution and plastics, there's definitely stress on coastal areas. We've seen mangroves disappearing and all these sorts of things. And although the problem is global, as was shown by the study in the Barents Sea, and also in this map that I'll go into in detail, a little more detail in a minute on the right from the IPCC, it seems that the manifestations are local. So it's not like sea surface temperature or atmospheric CO2, where we have a unidirectional trend where everything is warming or everything is increasing. The dynamics of the ocean mean that some areas might be seeing more biomass, some areas might be seeing less biomass. Not only that, they might have the same biomass, but it's a different species or a different community. And Heather will go into details on what that might mean later. But it's really, we have to look at the local scale to try and see the expressions of this broader change. So this is a panel just from the IPCC AR6 working group in chapter 2. And this middle panel, sorry if it's a bit small, but this is showing the trends at the global scale for different areas. So you can see the Arctic is seeing an increase in chlorophyll, the gyres potentially are seeing a decrease. And it really depends where you are as to what sort of features you would expect to see changing over the next 10 years and have changed over the last 20 years. So just in summary of the whole talk, the oceans, we can't stress this enough through all of these talks, is fundamental to how the Earth system functions, and we have to look after them. Ocean color is our only window really into the base of the marine food web and the marine ecosystems. So we have to, it's the only way really to monitor the microscopic plankton at the synoptic scale. And to examine this at climate scales, we need a very carefully merged record from multiple ocean color satellites. And that record has to meet the requirements of the climate community laid out by organizations such as GCOS. And basically now we're at the stage where we just need to use those records and really start asking good questions. So whether that's looking for forecasts of change to come by looking at what changes we're already observing, or if it's prioritizing regions that might need more protection because we see that they're changing very rapidly. or if it's identifying proxies or indicator metrics where we see something in the ocean color record that might be representing a much bigger ecosystem change. But now it's basically, we can say the data is available, it's growing as we continue to make the record longer, and it's over to the community to ask the right questions. So thank you very much, and I'll just leave that there.
Thank you, TJ, for this really wonderful presentation. Marie-Helene, are you there?
Yes, I am.
Perfect. I've already introduced you before at TJ's lecture, and I thought now would be a good time before we go on to Q&A for you to tell us more about the activities of ESA on the topic.
Sure. Thank you very much, Gemma, and apologize for the small technical issues encountered, but let's do it now. And actually, Thomas mentioned quite a few elements that you will find again in my introduction. So it was very nice to hear. I will share my screen and tell me if you see the presentation in the right.
Oh, no, we see your presenter mode.
Okay, so we will. Now we should be okay.
I lost your screen.
Okay, so let's try again. Where I am. Okay, share. Okay, let's share the screen and do it again. Now, what do you see?
Still the presenter mode.
And now?
Yes, perfect.
we will manage. Okay, good afternoon, good morning, everyone. Very nice to be here. Thank you for the invitation. My name is Marie-Helene Rio. I work at the European Space Agency, the Earth Observation Center in Rome, Italy, called ESBRIN. And I will do a very short introduction on this module 9 on ocean ecosystem and climate, ocean color and climate, by providing you an overview, very quick, obviously, of the activities that we have at the European Space Agency on the topic. And of course, as it is very short, don't hesitate maybe to get the slides, go to the links I put in the slides to the different projects. And at the end, I also give my email, and so you're very welcome to contact me for any questions. So the European Space Agency, okay, here we are, together with European and global partners, contributes to develop world-class Earth observation systems to address scientific and societal challenges. And here you can see the very complete set of Earth observation satellites that we have currently operational, being developed or being prepared. So it's actually 14 satellites operational as of today, with different lines, the science missions, the operational mission with the European Copernicus initiative, and the meteorological mission. When we look at ocean, a number of satellites are used to monitor the ocean from space, and providing information on many different variables that you can see here using different sensors, passive and active sensors. And so we have a number of parameters that can be used to study and to monitor, to further understand our ocean ecosystems and ocean health. So one of these parameters, of course, is ocean color. It's not the subject of the webinar today, but together with all other parameters, again, we have all the information or we have a substantial set of information needed to do science and to have a look into these key issues regarding ecosystem and climate. So to exploit this large amount of data, we have a number of programs ongoing, and the first one you already heard about in Thomas' talk is the ESA Climate Change Initiative, which was launched in 2008, so 25 years ago, in response to the UNFCCC requirements for having a systematic monitoring of the climate system. So through this program, what is being done is to develop the data records of key components of the climate system, which are known as essential climate variables. So there are 26 ECV, so essential climate variable projects ongoing, 2 budget closure projects, and a climate modeling project and data support project. So all the data you can download at the link below and find more information also there. Another important program that we have is the Geoscience for Society, a program which has a number of objectives that you can see here. In particular, fostering scientific excellence. So this is done through the development of science project where we address the needs of the scientific community in terms of new observation, new algorithm development, in order to tackle grand science challenges. A second important component, important objective is to pioneer new Earth observation applications. So here the objective is to really foster the use of remote sensing to develop new and reliable Earth observation application to support international environment and development policies and maximize public benefit of the Earth observation system. So the numerous projects that we have, they are gathered here into what we call the ESA Ocean Science Cluster. And I put here on this slide the names for the different projects that are running, actually more than 30. And among those, I put here in red the one which which are based, not only, but which are based on the use of ocean calorimetry data from the different satellites. Those different projects, we have also further clustered them into different topics, of course, which are linked one to each other, but we can identify main topics. And so we have projects looking at ocean health and biodiversity, project on coastal ocean, land-sea interactions, projects investigating operation dynamics, including Earth interactions. We are also looking at ocean extremes, at the ocean carbon cycle, and at the oceans following the Earth and climate system. So among these projects, an important point also is that 3 years ago, so in February 2020, we launched a collaborative framework between ESA and the European Commission called the ESA Earth System Science Initiative to really start launching projects in a coordinated manner between ESA and the European Commission. And we have defined a number of priority topics for which flagship initiatives have been created, and flagship projects are being started to launch collaborative activities. And among those, we have a flagship initiative on biodiversity and another one on ocean health. So both of these initiatives, of course, ocean color data are key inputs. So I will go very quickly through a number of those projects. It's not exhaustive. There are many different projects. You can have a look at them. I was giving the link on the slide with the Ocean Science Cluster, there is a link to the EO Science for Society website where you can browse per project and find more details on the objective and the link to the project website. But very quickly, so in the biodiversity flagship initiative, we have a project on coastal biodiversity, which aims at using satellite data to develop essential biodiversity variable for 3 different ecosystem types, so the intertidal, subtidal, and pelagic coastal ecosystems, a strong link with early adopters around the world to demonstrate the impact of these new products for specific use. In the biodiversity flagship initiative, we also have a project dedicated to open ocean biodiversity. It's called BOOMS, and the objective of this project is to develop dynamic seascapes from the use of satellite data, and again demonstrate how these seascapes can be used for a number of scientific investigation, but also for a more applicative for application, like the mapping of marine protected areas, linked to fisheries, and so on. In the Ocean Health flagship initiative, we have launched a set of projects. One is dedicated to marine heat waves, so how we can identify and monitor marine heat waves from satellite data. In this project, Marine Heat Wave Atlas is being developed, also looking at the 4D reconstruction of such events. And a number of scientific studies are being done to understand the impact of such extreme events on the marine biogeochemistry and ecosystem. In this ocean health set of projects, we also launched a project dedicated to acidification. So our satellite data can help monitor acidification in the ocean. And in this project, global dataset of the different parameters of the marine carbonate system are being developed for a period starting 1985 until today. The map shown here were developed in the previous project, Ocean Soda, and in the ongoing project, the team is working on improving the spatial and temporal resolution of this data, going 8 days and quarterly. And here again, one objective is not only to develop these new products over a long time period, but also then to understand how acidification, in that case, is impacting marine species in marine ecosystem, and also characterize how these ocean acidification events impact the ecosystem when they co-occur with other stressors as warming, eutrophication, or deoxygenation. The mapping of marine carbonate parameters is very important in the frame of ocean health, but also in the frame of better understanding the ocean carbon cycle. And we have a number of projects. We have had a number of projects in the tackling the different components of the ocean carbon cycle. So here I put a number of projects that you can see. I've been tackling the different pools of carbon in the ocean, but also a number of processes as primary production, export production, and exchanges at the interface between air, sea, and land sea. This nice schematic that you already saw in Thomas' presentation was done in the frame of the BICEP project. It's one of these projects, which was dedicated, still running, actually dedicated to the biological carbon pump. And so the estimation from satellite data of the different pools of carbon in the ocean. So the project is coming to an end at the end of summer. it has developed a data set of, from 1998 to 2020 at 4-kilometer resolution, monthly global of different products, particulate organic carbon, inorganic carbon, dissolved organic carbon, net primary production, phytoplankton carbon, export production, and so on. So all these data sets are available. You can go on the site, on the project website, and you can have a look at them. Another project which is still running is looking at net primary production in a specific case of upwelling system to advance our understanding on the link between net primary production and wind-forced upwelling, combining satellite with in-situ data, ocean circulation modeling, and climate indices. To look at what happens at the interface, we also had a project, the Syllabio in the Baltic Sea, whose objective was to develop methods for assessing carbon dynamics and eutrophication in this specific area of the world, and also calculating total oceanic carbon load from the 8 biggest rivers of the Baltic area. We also have had in the past and still running some projects looking at the fluxes. So the ocean flux was dedicated to quantify air-sea exchanges of greenhouse gases. And we also have this AMT for ocean sat flux project with on board an instrument on board the Atlantic Meridional Transit cruises to measure gas exchange. We also have, coming to an end, a project looking at extreme wind events, and I mentioned it here because it had an interesting case study linked to the ocean carbon. So in this project, a new data set winds was developed using the many different instruments that we have in Synergy that provide information about wind. So radiometer, scatterometer, and SAR, and an atlas of extreme wind events was developed, collocated with measurements of the underlying ocean environment. So each time we had a storm, so a tropical cyclone, a polar lows, or hurricanes, the project has been looking at what happened at the ocean, at the surface of the ocean in terms of temperature, color, salinity, and so on. And specific work has been done to understand the impact on the gas exchange and CO2 fluxes. And this is an example for Hurricane Alex in summer 2010. And this was found in the specific case that the storm induced a net reduction of oceanic outgazing by around 6%. And so the idea now is to upscale this study on a single case to the ensemble of tropical cyclones and storms that we have in the Atlas, to have a global view of a long time period of the impact of such extreme events on air-sea CO2 fluxes. So all these projects on ocean carbon, we had a very interesting ocean carbon from space workshop last year in February, where all these projects were presented, and many others. This was an international workshop, very successful, out of which a number of recommendations arrived on the current status and priorities for the next decade to further understand the ocean carbon cycle and how we monitor it from space. And this recommendation and this white paper that was issued out of it was actually also one of, so the workshop and the paper were actually deliverables from the CEOS, so Committee on Earth Observation Satellites, which is, of course, ESA is one member on this committee, and together with other space agencies in the world, we are trying to cooperate to make the best out of satellite data for Earth observation, and we have dedicated topics on ocean and aquatic carbon. So the workshop and the white paper were part of the work plans for 2023, and for the next work plan, so in the next 3 years, we have included other deliverables. So we plan to contribute or to develop an aquatic carbon roadmap in these 3 years, and also we have planned a blue carbon workshop that will take place next year in May. I also would like to mention that ISA is part of the International Ocean Color Coordinating Group, which is an international committee of experts comprising representatives from the national space agencies and research scientists for aquatic radiometry reserve community. The objective of the IOCCG is to promote the development and application of science and technology underpinning remote sensing of ocean color across all aquatic environments, so not only ocean, but coastal and also inland. So through coordination, training, and so on. And in that context, I think it is going to be very useful to all of you who want to know more about ocean color radiometry, to have a look at the resources pages of the IOCCG, where you can find a number of publications and reports which are very, very, very useful to further understand what can be done with ocean color radiometry. Finally, we have a number of projects upcoming. In the pipeline, we have 2 projects that should be kicked off very soon, I guess after summer. One, following the Ocean Carbon from Space workshop recommendation, we have launched a new project on ocean carbon cycle with the objective to produce an integrated budget of pools and fluxes of carbon in the ocean, so primarily based on satellite data, and also, of course, related in situ and models to reduce uncertainty and fill the gaps. And we also have another project that will advance our scientific understanding on the impact of multi-stressor events on ocean health, intended as a capacity of marine ecosystems to provide their services. So these 2 projects will last 2 years, and so they will be starting very soon. And finally, we have a project which is still to come. We are opening the call in the next weeks, actually, and this will target coastal blue carbon. So in this project, the objective will be to develop new methods to further improve the estimation of carbon stocks from remote sensing data for major blue carbon coastal ecosystems, so in particular mangroves, salt marshes, and seagrasses. And I think I'm already running out of time. I wish you a very successful, fruitful webinar. Again, here is my email. You are very welcome to contact me for any question to get further information on our program and projects. Thank you very much.